Good morning, everyone. Welcome to our Ask the Doctor webinar tonight. My name is Shelley Korean with SkyPass Foundation. I am joined tonight by my co-host, Dr. Shilpa Chitnis, movement disorder specialist at UT Southwestern, who also serves on our board of directors here at SkyPass Foundation. A very warm welcome to all of you. We are excited to introduce you to our guest for tonight, Dr. Kathleen Shannon. Dr. Shannon is the chair of neurology at the University of Wisconsin. Dr. Shannon, we are excited to hear from you today. Just a little bit on Dr. Shannon before we jump into the session tonight. Dr. Shannon, chair of neurology at the University of Wisconsin. She attended Northwestern University and Rush Medical College and then completed her training with a fellowship in movement disorders at Rush. Her clinical interests include the broad spectrum of movement disorders with special interest in Huntington's disease and Parkinson's disease. She has a long history of clinical trials research, as well as an interest in the potential role of gut-derived sterile inflammation in the genesis of Parkinson's disease. And as you guys know, our topic for tonight is biomarkers, inflammation, and repurposed therapies in PD. I know a lot of you have questions about the role of the gut microbiome and inflammation and other GI issues, and we'll be addressing all of that in our time together. This is going to be an interview style webinar, so Dr. Chitnis will be interviewing Dr. Shannon on our chosen topic, but we want to keep the conversation with you going as well. So please feel free to interact with us throughout the webinar. If you look at the bottom of your screen, you'll see a QA box. You can put all of your topic related questions in there, and any other commentary can go in the chat box. And with that, Shilpa, I think we can jump right in.
Yeah, thank you, Shelley, and welcome, Kathleen. It's great to have you. Telling Shelley the other day that I'm surprised that we didn't get you sooner, because you're such a great movement disorder doctor and such a great leader as well, and really inspiring person for women who want to take on leadership roles. So I'm really excited that you're here. So so what I want to do before we delve into a lot of the biomarker stuff and the work that you've been doing, I wanted our audience to get a sense for some basic stuff. So I wanted to ask you if you could explain to us what is, you and I are very familiar with this, but what is the Bragg's hypothesis, really in layman's term, and what would you think is the significance of that?
So that's a really great question to start with, and the answer goes back kind of a long time. So the main focus of the cause of Parkinson's now has to do with a protein called alpha-synuclein. And in 1912, the pathologist, Dr. Louis, noticed that people with Parkinson died with Parkinson's disease. So pathologists are doctors that specialize in dead people. So that the people who had died with Parkinson's disease had these what are called inclusions, which is clumps of protein in the brain cells in the area of the brain where the dopamine is produced, called the substantia nigra. We didn't know what that protein was till 1997, when a genetic discovery showed that it was a protein called alpha-synuclein, which is present throughout the nervous system. It's a very common protein, but that the cells that are affected in Parkinson's disease will accumulate this protein and then die. And so it's been kind of thought that it's the synuclein protein causing all kinds of the normal processes of cell function to go haywire that then leads to their death. So the next part of the story really is about 2006, when Heiko Brock, who's another doctor specializing in dead people, a pathologist, who've done a lot of work in what the kind of tombstones of Alzheimer's disease are. In other words, what are the markers in the brain of Alzheimer's disease, kind of moved on to Parkinson's disease. And he described that the changes in the brain, the protein accumulations, don't start in that dopamine area. They don't start in that area that we're so concerned and attentive to, the area where the dopamine cells are, but rather it starts lower down in the brain. And then in a later publication, he proposed that in fact, the abnormal proteins first starts to accumulate in the intestines, especially the stomach, the esophagus, but all throughout the intestines. And then that it spreads through the nerve connections between the intestines and the lower part of the brain, and then spreads up the brain, eventually causing the dopamine cell death, and then even farther up the brain to cause some of the late stage changes. So that's called synuclein spread. the other interesting thing, part of that story was that there were a number of people who'd had fetal brain cell transplants for Parkinson's disease. And people who died after those fetal transplants, it was clear that the fetus cells had the same protein that the brain cells did. So that was further evidence that synuclein, that protein, can spread from one cell to another. So if the cells are connected to each other, the protein can spread. And that's called the Brock hypothesis. And so it really was that one of the things that led to our particular research on the intestinal system and whether it's involved in the production of Parkinson's disease. So that's the Brock hypothesis. It's controversial, actually, because it may be that not everyone follows that same pathway from the intestines to the brain. And certainly we can see changes, the same kind of tombstones, the synuclein accumulations in the nerves for the olfactory system, for the sense of smell. But kind of the interesting thing about those two discoveries, one, that the synuclein abnormality can start in the intestines, and two, that the synuclein abnormality can start in the nose, is that we know in people with Parkinson's disease, when we diagnose them, they often have a history of constipation and or loss of sense of smell. So there might be actually symptoms that go along with those accumulations of protein in those areas too. So it's very interesting, but somewhat controversial still.
Yeah, thank you. This is really great. And, you know, even it's 2022 and, you know, patients say, you know, I got a brain MRI and my neurologist diagnosed me with Parkinson's disease. And I said, oh, I need to know that because I haven't learned that yet in fellowship, that you could get a brain MRI. Although there, you know, we know that there are some imaging signs now that you know, could be suggestive, but still we, this is a clinical diagnosis. And so do you think that, you know, and biomarker research, you know, has been going on for, you know, how long, decades. And yet we haven't really found one. So do you think that there is a possibility that we are going to find a biomarker so that we can think about, you know, premotor and preclinical stages of Parkinson's disease? And then do you think we'll be able to develop a test, you know, to diagnose, let's say someone's having a routine colonoscopy and you just get a biopsy and you know, is that something that can happen, and would this test be validated?
So one of the big problems, you've just hit the nail right on the head, is that these markers we see are in pathology in dead people. And we don't really have a way to measure the disease onset or the spread of the disease in living humans. And so the search for biomarkers, both imaging biomarkers and biopsy kind of biomarkers and blood biomarkers and spinal fluid biomarkers, is to try to find a way to identify the disease earlier, to identify the disease more accurately, to study how it spreads in different people and what kinds of things contribute to that spread, and then also potentially to demonstrate an effect of an experimental treatment. So in Alzheimer's disease, there is a scan that measures protein, in the abnormal protein in Alzheimer's. There are two, one's called amyloid protein and one's called tau protein, and they both increase, and there are markers for both of those. And they can be helpful perhaps in understanding if experimental treatment is doing what you think it is, which is clearing that abnormal protein, for example. But we don't have that. There are some biopsies. So one of the side effects of our research was not looking for a biomarker, actually. Our research was looking to try to understand the cause of Parkinson's. And let me just explain a little bit what we did. So we were interested in whether inflammation that comes from the intestines could possibly drive a degenerative process in the brain. And the reason we were interested in that was really several things. One, we know that Parkinson's disease is probably partly genetic and partly environmental. So the biggest surface in the body that can be exposed to an environmental agent is in fact the gut, which stretches out the size of a tennis court if you stretch the whole thing out. So it's big, it's big. And you think of that as your insides, but actually it's your outsides. You're like a donut. It's a hole in your middle, that's the intestinal tract. And so it's a very, very exposed to the environment, things you swallow, et cetera, and even things you breathe, but then get in your saliva and swallow. The second thing is it's really important for protecting you from bacteria and toxins, et cetera. It's a very important role there. It's very important in the immune system. So the system that kind of monitors and treats invasions, especially bacterial or viral or fungal invasions. And the other thing is the intestines are called the second brain because there's a huge number of nervous system cells that line the intestine. That's how food gets from one part to the other, is that the nerves supply muscles which propel the food down. It's not by gravity. So there's an incredible exposure, very, very close, within millimeters of the intestinal contents are nerve cells. And those nerve cells connect directly to the first nerve cells that Dr. Brock explained are affected in the brain. So there's a reason to think that there could be either spread of the protein, but also some other effect. And so, and then the other thing that we were interested in, there's a compound called lipopolysaccharide. It's a toxin that lives in the wall of bacteria that live in the intestine. So E. coli, a common bacteria, but most of the bacteria in those families produce this toxin. And the reason the toxin is interesting is if you inject that toxin into the brain, you get Parkinson's disease in animals. You get an experimental model of Parkinson's. So we thought maybe that exposure to that toxin could be driving an inflammatory process in the brain. So that's why we studied what we studied, not because of the synuclein. The synuclein was kind of a side project, and we did find that people with Parkinson's disease had synuclein in the intestinal biopsies. So we collected both stool and biopsies, and that's because bacteria live on the surface. They create a little like city living on the surface, and those bacteria can be different than the ones actually in the feces. So as long as we had the biopsy tissue, we thought, well, let's just look at synuclein. So we found that the Parkinson's patients had synuclein, and the control subjects who didn't have Parkinson's didn't. We also found that people who'd had biopsies years before they were diagnosed with Parkinson's also had that protein already in the intestines. Now that work turned out to not be replicated widely. So a lot of people found those changes in Parkinson's. Other people found the same amount in healthy people. And so that has not emerged as a good biomarker for predicting. So we thought, wow, if this is accurate, everyone needs to get a colonoscopy by the age of 50, which is a perfect time to see if they've got this protein, but it turns out not to be accurate enough. There is a commercial biopsy, so that's skin biopsy, that also identifies synuclein in skin. And that's being used in clinical trials, and it's available as a diagnostic test. The problem there is that, as you mentioned, it's a clinical diagnosis. So we diagnose it based on the history and the physical examination with a pretty high degree of accuracy. It's not perfect, but if you follow people over time, it's pretty darn good. And the thing is that synuclein isn't only seen in Parkinson's disease. So there are other conditions, Lewy body dementia, multiple system atrophy, something called primary autonomic failure that also involves synuclein. And so finding that protein in the skin doesn't necessarily mean Parkinson's disease. It means something in that family of diseases. So I haven't ordered that test. I don't plan on ordering that test. The other people have looked at the salivary glands, because the salivary glands, so you do stick a big old needle in there and pull out some tissue. It's gruesome to look at, actually, but that's not going to be something that's useful, I think, for the general population. So,
Yeah,
so far, no good
absolutely.
biomarker.
Yeah, absolutely. And then the Michael J. Fox Foundations, has this whole PPMI project and been trying to look for biomarkers for a long time. Thank you. That's really great. So let's talk a little bit about, you're not a gastroenterologist, but because you've done research in this area, maybe about the normal, human microbiome, and basically, is it the chicken or the egg? Is it, the alteration of the microbiome causing the inflammation? Is the inflammation causing the alteration in the microbiome? So if you can, comment on that.
So the microbiome, our entire, all of our surfaces are covered with bacteria. And the bacteria are important in maintaining health. You have to have them there, and you can develop problems if you take antibiotics and get rid of the bacteria, for example. And the intestines are very richly supplied with bacteria. You're born in utero, you don't have any bacteria in your gut, but as you come through the birth canal, you start to get populated with the bacteria in your mother's birth canal, in the vagina. Babies who are born by C-section actually have different microbiome than babies who are born by regular, normal, spontaneous vaginal delivery. And then you accumulate a stable microbiome over a period of years, kind of the adult microbiome. And just to give you an idea of how there are billions of bacteria in your gut, and half of your stool by weight is bacteria. Half is bacteria. So it's really important, and bacteria are important for kind of tuning up the immune system in the gut, and they're important for the gut health. So for example, there are people who take antibiotics for life-threatening infections who then develop a special kind of inflammation in the intestines, and a different kind of bacterial population is very serious. So maintaining that bacteria is really kind of important for health. So what we know about the microbiome in Parkinson's is, first of all, and there are lots of differences. So depending on where you live, so people who live in Africa, for example, have a completely different diet than we do. They have different kinds of bacteria than people who live in the United States. So there are differences across the world. But basically, there are bacteria in the intestines that are anti-inflammatory, so they reduce inflammation. And there are bacteria in the intestines that are inflammatory, so they produce inflammation. And what virtually every microbiome study has found, and the way we do those studies is you collect stool usually, and it has to be collected, especially the bacteria that live in the gut die in oxygen environments, oxygen-rich environments. So it's a special kind of collection. And they don't culture very well. So it's not like a urine culture where they just put it on a dish and look at the bacteria. What they do is they analyze the bacterial genes, and then based on which genes, they determine which bacteria are producing or in there. So that's kind of a genetic analysis. But basically, every study that's looked at microbiome in every country of the world, and there are more than 24 now, have shown that people with Parkinson's disease have more pro-inflammatory bacteria than people without Parkinson's. So they have more inflammation in the gut. And we've actually shown that there's inflammation in the gut wall and inflammation in the body associated with those bacterial changes. But the real question, of course, is could having Parkinson's disease make your bacteria inflammatory? So which comes, as you said, chicken or the egg? Is it the bacteria that cause inflammation that cause Parkinson's disease? Or does Parkinson's disease cause a change in the bacteria that makes them more inflammatory? And there's a little bit of a suggestion of evidence in that there are some syndromes that we consider prodromal Parkinson's, so changes that occur that aren't specific for Parkinson's, but are suggestive that the person might develop Parkinson's. And the most obvious of these is something called REM behavior disorder. So that's the tendency to act out your dreams, to kick, punch, fall out of bed, for example, when you're having a dream. And people with REM behavior disorder have a similar change in the bacteria. So they have more pro-inflammatory bacteria compared to people without REM behavior disorder. And so that might be a clue that the inflammation in the bacteria comes before the diagnosis of Parkinson's, but it's still not entirely clear.
That is very, very excellent. Also, just off the truncal vagotomies, like, the studies that truncal vagotomies to see if that would kind of, I guess, prevent you from developing Parkinson's disease. Is there any premise to that?
So the other kinds of research that we rely fairly heavily on is what's called epidemiology, which is people who have Parkinson's disease versus people who don't, what kinds of things in their lives might differentiate between the two. So there are studies in the old days, before we knew that ulcers were caused by bacteria, we thought that ulcers were caused by acid secretion. And so they were treated by things like drinking cream and milk, et cetera, but also by snipping the nerve that goes to the gut, which produces the acid secretion. And that's called vagotomy. And there are some large European databases of people with who've had vagotomies and with good connection to the medical record, suggesting that certain types of vagotomy reduce the risk of Parkinson's disease. There are other epidemiology studies showing that cigarette smoking reduces the risk of Parkinson's disease, and drinking a lot of caffeine reduces the risk of Parkinson's disease. And then there are epidemiology studies that show that inflammatory bowel disease, so things like ulcerative colitis or Crohn's disease, which are issues with the intestines related to inflammation, that having inflammatory bowel disease increases the risk of Parkinson's too. So that kind of makes the connection between gut inflammation and disease, and that certain treatments which reduce inflammation as a treatment for inflammatory bowel disease reduces that increase in risk. And that's a very interesting kind of observation as well. So there are a lot of these kinds of observations that seem to support this kind of theory about the genesis of Parkinson's, but it's still all very preliminary and has not really led to a treatment. So people will say, well, should I be on probiotics? Should I change my diet? We do know that diet, we now know that diet and a Mediterranean diet will change the bacteria to a more anti-inflammatory bacteria. So Deborah Hall presented that at the Academy of Neurology meetings this year. And there's another similar poster for that. And we also know from epidemiology that people who follow a Mediterranean diet have a reduced risk of Parkinson's disease. We don't know what happens after you have Parkinson's if you change to that diet, whether you can influence the course, but there certainly is evidence that an anti-inflammatory diet might reduce risk and also change the microbiome in a favorable way. The probiotic story is a little bit more complicated because it turns out that some bacteria that are in probiotics actually will break down the levodopa that we use to treat Parkinson's in the gut, so it won't get into the brain. So it's not clear whether probiotics are actually good or harmful for people with PD.
Yeah, no, very good. And just to follow up on that, people are looking at NSAIDs, non-steroidal anti-inflammatory drugs, as well as statins to think about, for either Parkinson's prevention or reducing the progression. So I guess that the jury is out on that as well.
So a lot of those ideas come from epidemiology as well. So especially the VA has a very large database of people who were diagnosed at the VA with Parkinson's disease, and then their medical records and what drugs they were exposed to. And so their data, as well as data of others, do suggest that people who take non-steroidal anti-inflammatory drugs, not necessarily aspirin, but the other ones, especially ibuprofen, might have a lower risk of Parkinson's. And then there's both preclinical research, so animal research suggesting that statins might slow the progression of animal Parkinson's. And also some epidemiology literature suggesting that simvastatin, for one, might reduce the risk of Parkinson's. But those things are all preliminary. And for every study that says yes, there's at least a study that says no. So the NSAID is the statin. So there are studies that say no too.
Yeah.
And one of the kind of jokes about epidemiology is that epidemiology only works in retrospect, that when you start to put people on these drugs and study and see if they have an actual effect on disease progression, you can't demonstrate that. So then the thing about epidemiology is you don't know why some people were on those drugs and why others were not, right? There's a reason people take statins. There's a reason people that take non-steroidal anti-inflammatory drugs. And so really to show something has an effect on the disease, you have to do a prospective study where you randomly assign people to the ibuprofen or something that looks and tastes just like it, and then follow them over time and see how the disease progresses. And we've been doing that. My first study to look for something that's neuroprotective was in 1985, the DataTop study. And there have been dozens of studies since then to try to prove any of these things has an influence on the disease progression, and nothing has turned up. No matter how much evidence there is in animal models, and no matter how much evidence there is in epidemiology, we still haven't hit on that one or more solutions to Parkinson's disease.
Yeah, absolutely. I mean, so, in our lifetime, we've been trying to look for a neuroprotective agent. Do you think there is, given how complex, Parkinson's disease has been, in some wonder whether you could call it a Parkinson's syndrome, with how complex is it? Do you think there's a premise for a neuroprotective agent? We still do not know what is the thing that triggers off the onset of this. You talked about the Bragg's hypothesis, but what is it that causes those things to happen? So given that, do you think that a biomarker is going to happen in our lifetime?
So
a neuroprotective agent is going to happen.
So I think that the two biggest hurdles in finding a neuroprotective therapy are, number one, no animal gets Parkinson's disease. Every animal model of Parkinson's disease is manufactured in the lab by either injecting a toxin or putting an abnormal gene into the animal. And they're even so there are animal models in mice and animal models in primates, monkeys, but we just, it's not the human disease. So that's a big hurdle. And then the second thing is, by the time we diagnose Parkinson's disease, there's plenty of pathology, plenty of cells that have died already, plenty of accumulation of proteins. And partly we're just not able to intervene early enough. And so there's a big push now to try to identify people who are going to get Parkinson's disease. There are kind of algorithms and models to try to identify people who are going to get Parkinson's disease, because then you could do a study. And if you had something to measure and it was early enough, you might be able to make an impact. But so those two things I think are major hurdles. And then the other thing, of course, is when you study the disease in animals, so many things go wrong with the cell. So the protein attracts other proteins to accumulate in the cells, and there's interference with how the genes are active. Other genes are active in the cells, and there are changes in energy production, the mitochondria. So there are just so many different things. And you can really only study one intervention at a time, two at the most, if they don't interact. And so it might be that what we need is some kind of cocktail that helps strengthen all those things, or something that intervenes in the primary process, the protein accumulation, in order to make any kind of impact. So we need to be able to diagnose it earlier, have something measurable that's real, and have a target that's effective. And so it's just complicated. We had a patient once ask one of my colleagues, well, how come we can put a man on the moon and we can't cure Parkinson's disease? And the answer to that is we know just where the moon is. That's math and physics. That's easy. But Parkinson's is just so complex. And it may be that people get it, for different reasons, right? It's maybe kind of the final event that happens as a result of some interplay of genetics and environment. And it may even be that it would be a different target in different people. So it's just enormously complex. Fortunately, unlike Alzheimer's disease, we have very good symptom treatments for Parkinson's disease. And, the experts are actually really good at managing the medicine to give people a really good quality of life. It's difficult at the end, as it is with any degenerative disease, but we have really good tools for making people's symptoms better. So that's a benefit for us.
Yes, absolutely. I mean, I have patients, from Louisiana when I was a fellow, that are still young onset. They're still functional, and they're some of them are obviously working with walkers and things like that. But they still have reasonable quality of life. So it's it can definitely manage between deep brain stimulation and all of the medical therapies and speech and physical therapies, big and loud, that we really can provide a good quality of life for patients. There is, no doubt about that. So just switching gears to a couple of drugs that, probably in study, and whether there is a hope for them to, to be real. So for example, exenatide, we've been really dealing with that for quite a few years now, and so any of those, glucagon like peptide agonists for disease modification, is there promise?
Well, so a lot of times in the early studies, and when you're kind of putting together a research platform to study a drug, the first step is always in animal models, and you find something that looks like it engages the target that you think is important. It works in animal models, and then you move it into kind of safety testing. But exenatide is an example, and isradipine is an example of medicines that are already FDA approved for an indication. And so the advantage to repurposed drugs, as you called it earlier, is that you don't have to do all this toxicity testing. So you know what the side effects are, at least in the population in which it's used. So you know what the side effects are, you know what the right dose range is. And so it gives you a jump start. So you save a number of years. But then you still have to test it rigorously. So you need a prospective study where you recruit enough people, and generally that's at least 200 to 300 people in each arm. So 200 to 300 people in the placebo arm, so an identical appearing intervention, and 200 or 300 in the active arm. And then you have to have something to measure. So we use things like the Unified Parkinson's Disease Rating Scale, which is just a severity rating scale for all the signs of Parkinson's, like how fast you tap your fingers and how well you walk and what your balance is, et cetera. And that's kind of how we measured. And then if we can show that the people on the exenatide, for example, had a slower decline in their function, then you might have hit upon a winner. But the fact is that in those large studies, nothing has emerged. So exenatide, isradipine, nothing has emerged as effective. So we just still haven't hit on the right formula there.
Yeah, and then just on the same lines, even inosine, their role for that agent.
Yeah, inosine's an interesting drug because it arose from an observation that people that had higher levels of uric acid, so that's the compound where really high uric acid levels are associated with gout, and there are medicines that reduce uric acid, and there are medicines, inosine, that increase uric acid. And so what they did was they got people who had lower than normal uric acid and Parkinson's disease, and then they randomly assigned them to this inosine versus a placebo to see if raising the uric acid would be associated with slowing of progression, since it seemed like people who had high uric acid levels had slower disease progression, but that didn't work either. So even though it was very thoughtful, well-designed, very scientifically rigorous study, it was a negative study.
And there are still people out there that admit patients for doing IV glutathione. And I know patients who just go, and then I don't know whether it's a placebo response, and they come back and they're like, oh, I'm better, Dr. Chitnis, my tremor is better, this better. And what do you tell a patient population?
So it's really important to understand the placebo response because it's fascinating. So the fact that you're getting some kind of intervention and you believe in the intervention actually causes real and measurable improvement in the symptoms. So the person feels better, the score can be better of any kind of rating scale. And so, and the thing that people often mistake about placebo effect is placebo effect happens in both arms. So the people getting the active treatment, they have placebo effect too, because placebo effect is just the response to taking something and thinking it's going to work. So they have placebo effect plus drug effect compared to just placebo effect. And so, and there's a theory that placebo effect has to do with dopamine, actually. So, because dopamine's in the reward system, and placebo effect may be kind of a reward kind of feeling. So one theory is that that Parkinson's disease patients are very susceptible to placebo effect because placebo effect might involve dopamine. It's interesting. So placebo-controlled studies are essential to know whether something is really making a difference. And glutathione is a perfect example. Glutathione has a very short activity in the body, very short, and so it's unlikely to be able to do anything for Parkinson's disease. It was kind of pitched by let's say a huckster, someone who wanted to make money off of glutathione. It's kind of sad because people, I think, are preyed upon by these snake oil salesmen. I would consider glutathione snake oil.
You're absolutely right. It's absolutely no doubt. So, is there anything on the horizon that you as an expert think has promise in PD, therapeutics? We talked, with Dr. Lang about even genetic therapies, therapies. Is there something that you look at it and say, this is really promising, and I'm going to follow this because this could be the real deal one day?
Yeah, I don't think it's possible to actually say. I do think that I'm interested. I would say that I'm interested in the newer approach, which is a synuclein targeting approach. Since synuclein is the common thing that unites everyone with Parkinson's disease. And we think it's critically important in the genesis of the disease. And so there are a couple different ways to approach synuclein. You can give a vaccine against synuclein. You can use what's called a monoclonal antibody that specifically targets that synuclein for the immune system to remove from the cells. Or you can find a way to ramp up the cell's own process for disposing that protein. And so those three approaches are all in clinical trials now. We're doing One of the studies, one of those ones that increases the ability of the cells to degrade the synuclein. And they're placebo-controlled, very well-designed studies. This is a phase 2 study, which is not all the way to a definitive study. But I don't know if you're participating in any of those trials.
No, not.
You know, and people will say, well, you know, are your patients getting better? But we don't know if the person's on placebo or active drug either. It's what's called double-blinded. The patient doesn't know, the treating doctor doesn't know either. And you only find out after all the studies done and all the data are collected, whether people are, who's on what. But I think that that's an interesting approach. It kind of similar to the approach in Alzheimer's disease where they're targeting amyloid. So that's the protein, one of the 2 proteins in Alzheimer's disease. The advantage in Alzheimer's disease, of course, is they have an imaging study that measures how much amyloid is in there. So you're able to see whether your treatment's actually having an effect. And we don't have an imaging study that tells us how much synuclein is in the brain. Although the Michael J. Fox Foundation has a big reward for anyone who comes up with that. And there's maybe some hint that there might be one in development, but we're not quite there yet.
Yeah, but the studies with like tau antibodies for PSP have not been that successful, right?
Correct. Yeah.
So
I mean, you can take a lesson from Alzheimer's disease. So you may be aware, your viewers might be aware that there's enormous controversy about the amyloid drug that was approved by the FDA. So the advisory panel said it wasn't proven to work. And several of the advisory panel actually resigned after the FDA approved that drug. And the reason the FDA approved it was because it did show change on the imaging marker. It showed fairly dramatic reductions in amyloid protein. It also saw a lot of inflammation in the brain too, like 40% was significant inflammation in the brain. But that wasn't really correlated with a significant improvement or change in the progression, but it still got approved. So very controversial. But does that mean, Kate, even if we do find a biomarker of synuclein in the brain, that might not be a reliable marker of improvement either? So it's a very interesting, and then the drug was very expensive when it came on the market, but then the Center for Medicaid and Medicare said they weren't going to pay that unless people were in a study. But it gave a lot of people, I think, false hope. And it's kind of a sad story. It's a good lesson for us because if we do find an imaging biomarker, it may not be as reliable as we think it would be.
Yeah, absolutely. And Dr. Shannon, this is great. I'm going to go and see what questions our audience have. Okay, so question number 1, have you been able to rule anything environmental out as a trigger? So whether it's insecticides, smoke, defoliants.
So I think that the strongest evidence for environmental exposure comes from the pesticide literature, actually. Partly that's because of the work of Caroline Tanner, who's an epidemiology expert and even older than I am. So she's been around Parkinson's disease a long time. And she works in California, and California has a zip code by zip code map of how much people are exposed to pesticides. And so they're able to kind of correlate the risk of Parkinson's disease with where they've lived their whole lives in California. And so there is an association with pesticide exposure that I think is fairly strong. But for most people, you might apply pesticides in your garden. And so the other thing I think that's sort of suggestive is there's a long time ago, Dr. Tanner described an association with well water drinking and living in rural environments, again, suggesting that something seeping into the well water that could be toxic and potentially a pesticide as well. Now there's a whole big lawyer thing where, if you've been exposed to paraquat, call this lawyer, you may be, that kind of thing. But the fact is that most people haven't been exposed to excessive pesticides that we know, and they still have Parkinson's disease. So probably there are multiple environmental factors. And Harold Klawans, who kind of found one of the first people to give levodopa to Parkinson's patients, who founded the movement disorders program in Chicago that I trained at, used to say it's going to be something as simple as a potato. And it's just, if you got the right genes, that potato gives you Parkinson's disease. And it's maybe just different stuff that shares some kind of common biological activity. It's just a tough nut. All the neurodegenerative diseases, we haven't found one exposure that's consistent across patients.
Yeah, so I spend a lot of time in southern Louisiana, and one of the things that people like to do there is fishing, and they use because there's so much weed in the rivers, they try to use rotenone. So they use a lot of rotenone, and then people eat the fish, and then there is a little bit of a higher incidence of Parkinson's in that population. And then the other thing that goes on in Louisiana is basically the welding. So there's lots of lawyers that go there to talk about welding and Parkinson's disease, where it is actually Parkinsonism, not really
not the disease, right,
or manganese related Parkinsonism.
Yeah. And the newer welding rods don't subject people to that toxicity. It's only the older welding techniques that did that. The problem is, once the lawyers get involved, the science all just goes away, unfortunately. But rotenone and paraquat are the two specific pesticides that have been kind of linked to Parkinson's. It's interesting.
Actually, there are more patient questions. The question came to my mind when you were talking about, field transplants, right? You were talking about how when you transplant and the transplanted tissue takes on the pathology of the toxic environment. And we know that it is not just the dopamine story, right? Parkinson's is not just dopamine, it's also so many neurotransmitters. Why are people still doing stem cell trials? Like why still there an interest in stem cell trials, given knowing what we know?
I have to say that I think that it's fairly uncommon for basic scientists to create such a following. Because basic science seems so unreachable and difficult to understand. And so for the population, I think, but the stem cell scientists made real inroads into getting people to rally around stem cells. And the idea is that these are immature cells and they could replace, restore, regenerate brains with neurodegeneration. The fact is that in transplantation for Parkinson's disease, you don't put the cells where the missing cells are. You don't put them in the substantia nigra. You put them where the dopamine goes. And so you cannot reestablish those kinds of connections that you would need to cure the disease, for example. And I think people have kind of pinned their hopes on this as being some kind of cure. But really what it is, it's a source of dopamine. So those are cells that secrete dopamine, and they're in the area where dopamine cells, the cells that respond to dopamine are, and so they should improve in the same way levodopa does. They should improve the symptoms of the disease, but you wouldn't have to take it orally because the dopamine is being produced by the transplant. And the fact is that, like much research, there were a lot of small studies that weren't adequately controlled that suggested that it was really these fetal transplants were very effective. Early stem cell transplants are effective. Once you do a study, a really well-controlled study, those things haven't been effective. Yeah. But what's kind of sprung up, unfortunately, is there are a lot of commercial stem cell treatment centers that pitch stem cells as the cure for basically everything. You go to the website, it's fancy, the doctors are wearing white coats, everything is clean and blue, really, really great. And then there are testimonials, but there's absolutely no evidence to support that. And the FDA just can't do a good job at shutting those things down. But they're really a whole new level of snake oil salesmen, snake oil stem cell salesmen. And one way that you know if someone's doing legitimate research is you don't have to pay for legitimate research.
Correct.
You pay when you go to a stem cell clinic. You pay through the nose when you go to a stem cell clinic. So unfortunately, even though they really are selling a lot of hype, it's better to invest in a healthy diet and exercise. So one of the interesting things that came up, so, I'm sure you're the same as I do. I am hawking exercise all day long. I know that my patients who exercise daily do much better than patients who don't. It's unequivocal. No doubt. I can walk into a room and know that a person's exercising just by seeing how well.
Absolutely.
And but that's hard to prove exercise because to prove it in a scientific way, you got to kind of get hundreds of people and randomly assign them to exercise or something that's not exercise, and then follow them along just like we would in a drug trial. But some interesting stuff that's come out from, there are these populations, you mentioned PPMI. So these are prospective populations who sign up when they're diagnosed and then get followed up every year for years. And so you know what kind of things they're doing, they're eating and they're exercising, and you know how their disease progresses over time. And a publication came out in February showing fairly definitively that people who exercise 150 minutes a week, it's just a little bit more than 20 minutes a day, had significantly slower progression of their disease over time than people who didn't. And that's a really strong argument for exercise being extremely important in the long-term outlook for Parkinson's, both because it makes you flexible, keeps you strong, but also probably has an effect on the disease itself. So, you know, investing in a personal trainer, health club, whatever. My patients really love the boxing related exercises, you know, rock steady boxing. It's hugely popular here. And I just love rock steady boxing because the trainers know, they understand the disease really well. And they also know how to work with people who are at various stages of the illness. So even later stage patients can benefit from that. So I'm a huge proponent of exercise.
Yes, absolutely. And I have no doubt, you know, just like you, have patients who, you know, I mean, you look at them and you're like, wow, you've had disease for 15 years, really, and you look really good, right? And that's because I know that the answer to that is because they get up in the morning at 5 a.m., go to the gym and work out. So there is no doubt. I mean, and then, I mean, you do believe that exercise probably releases some nerve growth factors and could preserve the neurons, right?
And the other thing about exercise, so we mentioned inflammation. And inflammation in the brain is very difficult to treat. We aren't very good at treating inflammation in the brain. But both an anti-inflammatory diet, so the Mediterranean diet, and exercise both probably have an anti-inflammatory property. So, I'm also pretty big on the Mediterranean diet for our patients too. Do you do that too? Do you recommend Mediterranean diet? Yeah.
Yeah, absolutely. Yeah, I mean, that's, you know, it was a perfect diet and perfect diet, but I think that the Mediterranean diet is, it's also very sensible and it, you know, makes
good
sense.
too.
It's
really good.
Absolutely. Yeah.
And lots of fruits and vegetables, so it helps with that constipation problem. That's such a nuisance.
Yes, and we can probably spend 2 hours talking about that.
Yes.
Okay, so what do you think about this is an interesting question, regular use of standard fleet enema usage to flush the buildup of bad stuff like synuclein and bad bacteria buildup?
So I'm not a proponent of regular enemas for that because I think that it disrupts the bacterial, good bacteria too. Fleets doesn't know how to pick the good ones from the bad ones. And also dependence on enemas makes your gut kind of lazy. So enemas have a place when you really get backed up, but it makes more sense to try to prevent constipation using, you know, fiber and Miralax. I'm a big Miralax fan. Prescription medicines if needed. I think one of the other reasons to kind of keep the bowels moving as well as possible is when people get backed up, I don't think they absorb their medicines quite as well. So getting a regular bowel movement is a really important thing for medication of Parkinson's too.
Absolutely. So any poop transplant research going on?
Yeah, there is some poop transplant research going on. It's hard to know how to do that. So who are the donors of the poop transplant? So there's one study where they selected like the perfect poo, people with the perfect poo to be the donors. And they did that by analyzing their microbiome, et cetera. And I know one of those studies, that perfect poo transmitted an infection actually in some of the recipients of the stool. So it's a little bit hard to judge that research. It's very, very early. And I wouldn't say there's anything definitive that's come out of that. Better to fix your own poo with the right diet and get someone else's poo.
Oh, goodness. Have you heard of that? I have a vision of that. Have you heard of using CBD oil for inflammation? Bought some CBD oil with 30 milligrams of CBD and CBDA. CBD oil?
So CBD, I just reviewed a grant for CBD. Was it in PD or I think it was actually in multiple sclerosis. It's hard to say. I think there's not good quality research on CBD, and there's not good quality research on cannabis either. And mainly because cannabis was inappropriately put into Schedule I with heroin, and so you actually couldn't research it. As a scientist, you couldn't have access to it. The federal government has a stash of marijuana somewhere. I once wrote a grant for the state of New York wanted to do a study because they wanted to get medical marijuana approved. And so I had to write a grant for them, and we had to use the stash and the federal stash of marijuana because it's illegal, right? It's an illegal drug, so you can't purchase it anywhere. So it's hard to know. It's really hard to know. I don't really have an opinion about CBD. I don't recommend CBD for any particular reason. Do you, Shilpa?
No.
It's got this aura of magic around it, I think. Like my sister thinks it works for everything, but it doesn't. It doesn't work for anything she thinks it works for.
Well, I think what I've heard from patients is that they feel less anxiety and they calm down, and their the tremors are better and they sleep better. And
but see, that could all be placebo effect too. Placebo affects all those things, right?
Yeah, it could be. Yeah, so,
but that's
whatever
fine.
I hear is
that's fine. If it's placebo effect or not, whatever it is, if you feel better, fine. So I put CBD in there's a category that I would saw the if it feels good, do it category, and that's in that. So that's like acupuncture, massage. Those are all in the if it feels good, do it category. So you try it. If you feel like it helps your symptoms, fine, you can do it. But that's not science. That's anecdote.
Yeah, absolutely. I mean, those are like ancillary therapies that you could use, but yeah. So Sharon Roberts says, I do not have the gene for PD. I have ulcerative colitis and take Humira. My daughter has Crohn's and takes Remicade. What are the chances that she will get PD?
Well, interestingly enough, Remicade is the one that's associated with the reduced risk of PD in people with inflammatory bowel disease. But it's so there's a bunch of genes for PD. Most people don't have the genes for PD, although in certain populations, like the Eastern European Jewish population has a higher incidence of genetic Parkinson's disease, for example, and people from North Africa have a higher incidence of genetic Parkinson's. But it's clearly some combination of genetic susceptibility and environmental exposure. I recently read that if you have Parkinson's disease, there's one study that if you have Parkinson's disease, your children have a 2% risk of Parkinson's disease. I usually tell people a 10% risk tops of Parkinson's disease if you don't have an identified genetic factor. So it's not really worth worrying about, actually. But people still, I see all the time people referred in because they've got some symptom and a family history of Parkinson's disease and are concerned that symptom might be Parkinson's disease. So a lot of what I do is reassuring people that it's not a very strongly hereditary condition.
Yeah, the only reason to
it's common,
absolutely.
It's not strongly hereditary.
Well, the only reason to think about genetics in patients is really if there will be some good genetic therapies that are directed against either LRRK2 or GCase modulators against GBA. Yep. So those kinds of things would be the reason to at least start thinking about genetics. But again, I mean, unlike the Europeans, we can't pay for genetic testing.
Well, so
it's hard.
And so I know a lot of people who've had genetic testing from 23andMe. I've done that myself. So for example, it tells you things like your earlobes are attached, but I already knew my earlobes were attached because I looked at my earlobes. It tells you which flavor ice cream you like.
Really?
Oh God, yeah. So there's always something new coming out of there. But then you get this report from them, and I had a patient come in and he said he had Parkinson's disease, and he said, "Well, I had my 23andMe and it says I don't have Parkinson's disease." Well, it doesn't say you don't have Parkinson's disease. It says you don't have the gene that they test, one gene for Parkinson's disease. So the gene PD study that's being done. And I've given people information about how to sign up for that study. Is a study for people with Parkinson's disease, and they have a virtual visit and they have genetic testing. They get a cheek swab kit.
Yeah.
And then what's nice about that is a genetic counselor gives them their results. So you not you get a report. It says this report says I don't have Parkinson's disease, and now you take to the doctor. But that's a really good study, and I'll give us a much better idea about the genetic forms of Parkinson's in the general population. I understand that they've had a lot of interest in that study, and they can't really accommodate everyone. But have you sent anyone to that study?
There are some patients who a part of that, but not hugely, I guess, and again,
So,
I mean,
the advantage to knowing that you have a genetic form of Parkinson's is you can predict the risk in your offspring, for example, and what you said. So it might turn out that there's a very specific treatment that might work in the genetic forms, because the genetic forms are in a sense purer than the non-genetic forms.
Yeah, but it's also really hard because, for example, like Sergey Brin, the Google founder, whose mother has Parkinson's disease, and he has lung too. But it doesn't mean that he will end up getting Parkinson's disease just at a higher risk, right? So that also puts you at a little bit of stress. You're gonna, sit your whole life. I mean, if I found out that I had a positive gene, I'd just get on the treadmill or the bike, and like you said, I mean, if you're going to invest the money, just invest it in these real things which will improve your health and probably maybe slow down the course of the disease. Let's see, one last question. So you kind of touched on that, but alternative methods that help slow down the progression: meditation, chanting, vibration therapy.
So that's that also goes in the if it feels good, do it category for me. Some of those things will certain of the symptoms of Parkinson's are very sensitive to your stress, especially tremor. So if you get anxious, you're going to shake like nobody's business. Even if you're on really good medicines, you still may shake, or your symptoms may still act up when you get stressed out. So to the extent that any kind of stress reduction therapy, meditation, chanting, any of those things kind of calms the nervous system down, that's good for symptom control. And learning mindfulness and relaxation training can be very helpful for people, as can treating anxiety. If anxiety, and people who are anxious often have poor quality of life with Parkinson's. And if you can get the anxiety under control, it gets a lot better. So partly sometimes that's medications for anxiety, and partly it's things like those that were mentioned. So anything that feels good, calms you down, is good. But it doesn't alter the disease progression as far as we know. It just makes the symptoms better.
Yes, absolutely. But I mean, you do believe that there are, based on what Dr. Jankovic has tried to find, that there could be, these three phenotypes of disease: one which is a, quite slower progressive, and another one which is a more malignant progressive, and then there is this intermediate one. And so if you're lucky to have had, I guess a milder progressive disease, then that's, that's just luck of the draw, I guess.
Yeah, and there are certain profiles. So people who have a lot of tremor often have a more slowly progressive disease. The tremor is annoying as heck, but it's a more slowly progressive disease. Women have a slower progression than men. So there's certain kind of profiles, but that still doesn't, you can still say, well, I'm a woman and my disease seems to be progressing a lot. So there's certain kind of generalisms, but when it comes down to progression, it's very individual, I think. And people who progress slowly in the beginning continue to progress slowly, and people who progress rapidly in the beginning continue to progress rapidly. So there's a certain, variant of the disease that you get that may be slower or faster. And part of that may be influenced by your gender or your whatever other treatments or genes or whatever, but it's hard to predict.
Yeah. And Kathleen, there is so much variability in how even movement disorder doctors, treat Parkinson's disease. So if you didn't train in the DBS era, or you weren't a big believer of DBS, and then you didn't refer patients, and by the time, we have a big DBS clinic, and we get these referrals for people, and I'm like, I wish you had been referred to me 5 years ago. I think I'd have been able to help you more, but now you're at a stage of your disease where your non-motor symptoms are impairing your quality of life, or you're falling and those kinds of things. So I'll still be able to get your dyskinesias to go away and, get maybe a smoother response, but you're not going to be able to have the kind of quality of life that you would have if you were appropriately selected within a good time window before, the non-motor symptoms raise their ugly head. So I think that, the quality of care that you get also does matter, I think.
Definitely. And I think that's a really important point about whether people get the opportunity to participate in DBS at a time when their disease will really benefit. But I also see, so I've got someone on my schedule tomorrow, and I looked at his chart, and he took levodopa for a week and he didn't like it. So now he wants DBS. Well, that's not a DBS candidate.
Yeah.
So part of it is you kind of expectation setting. I rarely have a spirited discussion with my patients, but I had another patient who had tried levodopa several times and had gotten quite severely affected without levodopa and wanted DBS. And I said, no, you have to have levodopa first. And she was so angry at me. And then she was miraculously better on levodopa because it's a miracle. It really is. It's a really good drug.
Yes.
So partly people think that, surgery is going to be and the other thing that people think is, if I didn't respond to the medicine, then DBS is my answer. But that's not true at all. DBS only works in people who respond to levodopa. And another reason why you have to have tried levodopa in order to be eligible for DBS is it only works in people who have responded to medicine.
No, absolutely. I mean, I have had a few patients that just could not tolerate levodopa. Yeah. I mean, whether it's nausea or whether it's diarrhea, somebody had, really bad gastroparesis, and they have, I think I've heard about 3 patients that, they looked very much like idiopathic PD, they had a number of years into the disease, and we did DBS, and it did improve their quality of life, and they did better than what they were doing because they were without anything. So,
but those people would have responded to levodopa too if they could tolerate it.
Yes, they would have.
I've never had anyone who I couldn't get on levodopa. Never, not once in many, many years. There's little tricks you can do. Like sometimes these really sensitive people. I learned this from Eric Alskog at Mayo Clinic. You started at bedtime. And what happens is you get the long duration effect. So they're better during the day. Correct. You add it all at bedtime. Right. And then once they're on a reasonable dose, you start moving it into the daytime. And that I've been very successful with that strategy. And people
really, that is
tried levodopa multiple. I learned it from Eric Alskog, and it's brilliant actually. And then you get them on daytime levodopa. And it's a slow process, and it requires a lot of kind of touch points with the patient, but you can get them on very gratifying. That's what happened with this lady I just told you about. She was so mad at me. I said, you got to have this. We're going to do it this way. And I just saw her again. It was remarkably better on levodopa. It's a wonderful story, actually.
Definitely. Now that Tygan is off the market, what are you using for nausea?
Well, it's a good question. I usually find that if you can give it at bedtime, let people sleep through it. One of the issues is when you first start the medicine, you're not getting enough carbidopa to block the nausea. And so some people will get sick, on a half a tablet 3 times a day, but you need at least 3 full tablets a day before you have enough carbidopa. And some people need more. So you can give them carbidopa, which is expensive.
Yes, you can give them Lodocin.
Yes. Yeah. And then also if they take it at bedtime and sleep through it, sometimes once they get enough on board, then you don't have to deal with the nausea. But I really have not had anyone who couldn't take nausea. Never.
That is a really good trick. I will definitely be trying that.
Try it. Really good.
I will try it. I'll let you know. Actually, we're trying to see whether we can get domperidone. There's lots of paperwork that needs to go, but I'm going to do that so that at least that would be a good because Zofran I don't think really works for these people.
Some people it works for, I guess. Yeah, so I did a clinical trial of domperidone way back when I was a fellow. And nice. But Janssen never got it approved in the US. And for a while it was pretty easy to get from Canada. You just had to send a letter and a prescription, and their credit card would be shipped. And then the customs kind of cracked down on that after a while. And it's much more hard to get now. I wish they would just approve it here. It's a good drug.
Yeah, absolutely. But every time I go to India, I at least get because I have motion sickness, and domperidone works like beautifully for me. So I got a huge stack of that. It's very cheap in India, and it's always there. You know, it's available. One of my patients said to me, "Why don't you give me some of your stock, Dr.
To put in the slammer for importing drugs, approved drugs. Yeah, you can import. The rule is you can import an unapproved drug for your own personal use. That's kind of the customs rule. That's how we got by with that for a long time with domperidone.
Yeah, no, absolutely. What is best for off episodes?
Well, I mean, the most important thing is to figure out why you're having an off episode, and then try to work towards that. And so one of the most useful things for people who have a complex response to medicine is a diary, where every half an hour, you just write down whether you're on or you're off, when they're having dyskinesia. And then, because I think for a person who's fluctuating, especially with bad off episodes, often you're so involved in the off when you're off that you fail to see the pattern. And so you can't describe the pattern to your doctor. And so a diary that you can say, oh, wow, so between 2 and 4 is when I'm really having most of my off episodes. How do we adjust the medicine around that time frame to reduce the off? So I think there's a lot to be said for diary keeping and people who are confused by their function fluctuating throughout the day and whose doctors are confused by the fluctuating function. And we do that a lot in clinical trials, looking at medicines that reduce off periods. Diary is like the #1 outcome measure. So reducing the number of hours off will get a drug approved if you can prove it reduces the amount of time off. And so I think getting a good understanding of that is the first step. And then kind of working to improve what you're identifying from the diary.
Yeah.
And then, so with the apomorphine injections, I don't use many of those. And Brija, which is an inhaled form of levodopa. Those are other options. They can be expensive. Apomorphine is very expensive, but so the Kinmobi, right? So there are things that that can be done. But again, you have to start with that pattern. What's the pattern? And then try to address the pattern.
Yeah. And then, you're talking about keeping a diary, and now just, the Strive PD, the Apple Watch, just got approved. And so, that's just another way of, I guess, selling more Apple Watches. But, I'm very much into technology, but I will say I completely agree with you that getting a good patient history or keeping a good diary is probably all that you really need. You understand that, machine learning and all those things, are good, I guess, but there's just no, in the history lies the mystery, if you can get a really good history, I guess.
I think it's worth hoping that in the future we have some kind of responsive drug delivery system that recognizes when a person is wearing off and then delivers a drug. I think that would be the ultimate benefit of a technology approach to PD. And maybe we'll get there one day. So I don't.
I mean, it's just
study, but the subcutaneous pumps look to be for people who are complicated fluctuators. One of my colleagues is doing a study here, and the study coordinator says it really makes a difference in bad fluctuators to have this pumped in levodopa, this new form of levodopa. So that might be. And potentially there could be a way where your watch would adjust your flow or something, because it would know that you're off. I mean, you can imagine that the future might hold some really, really sophisticated technology for delivering the medicine in a responsive way.
I think it would just like, like we're doing adaptive DBS, right? So, I was just telling a patient that at some point Dr. Chitnis could be obsolete because I wouldn't need to sit and do programming.
Never be obsolete. You're too special to be obsolete.
Oh, that's very kind. You just made my day.
Yeah.
Well, Shelley, how are we doing? I think we can keep talking and talking, but I guess we're limited by time. But this is phenomenal. Very, very excellent. And Shelley, go ahead. Take it away.
I second what Dr. Shannon said. And I think all of us do. Dr. Chitnis, you're too special to be obsolete.
Exactly.
Dr. Shannon, thank you so much. You're so fun and just such a wealth of knowledge. And this was such an interesting conversation. There were a few questions about swallowing and acid reflux. Any suggestions on how to deal with that don't include taking a proton pump inhibitor?
No, I don't have any ideas about reflux that don't involve taking. And the problem with proton pump inhibitors is they reduce the acid so much that you can't tolerate any acid anymore. You get used to just so low acid. and they're really hard to get off of. I don't know if Dr. Chitnis has any ideas about reflux. I mean, sleeping with the head of the bed elevated, those kinds of things are useful because a lot of reflux is at night. But I think my patients usually end up on proton pump inhibitors. Do you have another approach?
No, I mean, pyridostigmine sometimes helps a little bit better at, you know, being like a prokinetic. So yeah, that helps sometimes. So
yeah, I haven't tried that. That's good. Interesting.
And any experience with curcumin or turmeric with black pepper? Is there anything to that?
Same old story, not studied. No scientific evidence. Feels good to it. It's delicious, right?
Right. I think there's only anecdotal evidence.
It stains your marble countertops though, so be careful with the turmeric.
I would say though, the anti-inflammatory benefit of turmeric is, during the pandemic, and we can end after this, is my husband came up with a concoction. He warms up water, he puts honey, he puts a little bit of turmeric, and he puts lemon and pepper. And, seriously, like all of us who have taken it in our family, I think we wake up feeling better. Like we were better. I don't know what it really reduces inflammation. Like I have rheumatoid arthritis and I'm in complete remission. But I always wake up sometimes feeling like really tired and feeling like my body's on fire, and it got better.
It's a delicious placebo, Shilpa. Delicious, right? Delicious and colorful placebo.
Oh goodness.
Well, thank you both so much. This was such a great conversation. I hope we were able to answer all your questions, and I hope you benefited from this discussion. This is being recorded. It will be put on our YouTube channel, so please share it with folks that you think will be interested, or if you want to watch it again, we'll be sending out the link to everyone that's on here. So thanks again, and Dr. Shannon, I hope we can chat with you again soon.
This was really fun, Shelley. I really appreciate, and Shilpa, I knew it would be fun because Shilpa's involved. I didn't know you before, Shelley, but it's really nice, really fun. Appreciate the opportunity.
All right, you guys, have a great night.
Thank you, everybody.
Thank you. Thanks, everybody.
Bye-bye.
Bye.